Hello everybody, my name is Stefanie Erdaams. I'm a PhD student at the University of Antwerp and a biomedical scientist at Novosanis. Today, I'll take you into the world of urine as an emerging liquid biopsy. Today, the way many diseases are identified, classified and treated is changing. We are moving from a one-size-fits-all medicine to personalized or precision medicine, especially in the area of oncology. The molecular profile of an individual patient and their disease influences the effect of a medicine. Biomarker diagnostics can help to target the right medicine to the right patient. Biomarkers can be detected on tissue, but very often obtaining enough tissue is an issue. It may be difficult to reach a tumor or get enough tissue sample. Additionally, tissue biopsies are expensive, invasive, and do not allow multiple sampling. They are single snapshots and do not address tumor heterogeneity. Despite the challenges, tissue biopsies remain the golden standard, even today in cancer detection. Researchers are continuously exploring alternative methods to detect cancer types. The use of minimally invasive procedures such as liquid biopsies and detection of circulating tumor markers in body fluids such as blood and urine is gaining interest. Liquid biopsies are non-invasive, address tumor heterogeneity, allow real-time monitoring, and are less expensive. The most often used liquid biopsy is blood, which uses either serum or plasma as a sample type. However, blood has several limitations that can be resolved by urine. Compared to blood, urine is absolutely non-invasive, can easily be collected at home, and does not add burden of substantial blood draws. Moreover, the collection of urine is not limited by the health stages of a patient and does not entail any risk of transmission of bloodborne pathogens. And the high volumes that can be obtained per patient address the limitation of low biomarker concentrations and allow multi-omic testing to detect a complete panel of biomarkers. Let's first have a look at urine as a sample type and how it's used in research. First, there is a range of terminology used in the field to characterize the different times and fractions of the urine. First morning, being urine collected at the start of the day. First void, being the first part of the urine flow at any time of the day, and mostly refer to the first 20 ml of urine. Midstream, being the urine collected after discarding this first part, and rending being used for urine collected at any time of the day and any fraction type. Secondly, there can be a division made during processing. Most of the processing protocols will first centrifuge the urine sample to divide the sample in urine sediment at the bottom of a tube and urine supernatant on top of that. However, some protocols will work with full void without a centrifugation as pre-processing step. Next to the fact that urine can be categorized in different collections and fractions, we will now look a bit closer into the content of urine as a sample type. Urine contains a lot of components that can be used as biomarkers for the detection of diseases such as cancer and urinary tract infections. Here we see that urine contains both cellular and cell-free components, but proteins, metabolites, and volatile organic compounds can also be found in urine. Even the concentration of metals in the urine can be used as a biomarker. Urine clearly is an emerging liquid biopsy. A quick PubMed search showed that in the last decade, the number of publications concerning urine and oncology, or urine and extracellular vesicles, or cell-free DNA, or liquid biopsy in general, are massively increasing. For urine oncology, there was almost a doubling of the amount between the 3 decennia so far, as you can see on these graphs. Several studies have shown that the use of urine as a liquid biopsy for cancer detection and monitoring is promising due to the ease of sampling and the high acceptability compared to blood and tissue. And most interestingly, all types of cancers can be detected. To provide a clear view on the potential of urine as biomarker source for cancer detection, we are performing a systematic review according to the principles of PRISMA. But for today, I would like to share some interesting results with focus on articles published in 2020 and 2021. Probably not a surprise that urine can be used as a liquid biopsy for urinary tract-related cancers such as bladder, ovarian, colorectal, prostate, cervical, endometrial, and renal cancer. However, urine can also be used as a sample type for the detection of thyroid, lung, liver, breast, pancreatic, and gastric cancer. First up, bladder cancer. For bladder cancer, voided urine cytology is still included as a standard of care as an adjunct to cytoscopy to detect high-grade tumors. regardless of the limitation it has, such as an overall low sensitivity. I'll highlight two biomarkers used for bladder cancer detection. First up, telomerase reverse transcriptase promoter mutations. They were a game changer for disease monitoring and early detection of recurrence. These mutations are not present in inflammatory or urinary infections, which is a current pitfall in some of the non-invasive assays today. Furthermore, they are being currently included in tests such as Urine Monitor and UroSeq. Next to that, several studies have showed the potential of microRNAs in the detection of bladder cancer. Some used a single microRNA, others a panel of up and or down-regulated microRNAs. All studies were able to show a sensitivity ranging from 72 till 90% and a specificity ranging from 82 till 90%. A vast offer of tests and kits are available for the non-invasive detection of bladder cancer, with each assay presenting its own advantages and disadvantages. No leading kit is currently being broadly used. A large part have been on the market for some time and in general present better performance than conventional cytology. However, these kits are not yet implemented in clinical practice, since none of them are fulfilling all the expectations in all points of the detection of bladder cancer. However, healthcare professionals could take advantage and use each kit according to the specific patient, context, and limitations in the detection of bladder cancer. For prostate cancer, on the other hand, many studies have been performed. One study showed the interesting fact that digital rectal examination can enrich the biomarkers in the first vote urine fraction. But all different types of urinary components have been shown to have potential as a biomarker for prostate cancer. The most important examples are the long non-coding RNA prostate cancer antigen 3, the fusion protein TMPRSS2-ERG, the methylation marker glutathione S-transferase P, and furthermore, a systematic review from 2020 showed 4 potential urinary microRNAs as a biomarker for prostate cancer. And even more interestingly, there are already 4 urinary prostate cancer tests available on the market today. Cervical cancer then. One of the major causes of cervical cancer are high-risk genotypes of the human papillomavirus. The use of urine as a liquid biopsy for HPV DNA testing was shown to be very promising because of the high correlation between urinary and cervical HPV DNA, the ease of sampling, and the higher acceptability by women. Van den Helden et al. Evaluated the utility of different urine fractions for methylation analysis for the detection of cervical cancer. They showed that methylation analysis performs excellent in all urine fractions for cervical cancer detection. The existence of a large variation in urine samples, both between individuals and between fractions, is demonstrated by the large range in DNA yield per ml urine as found in this study. Nevertheless, valid methylation test results were obtained for nearly all individuals and urine fractions. Ritter et al., on the other hand, performed a study on different gynecological cancers. He chose ovarian and endometrium cancer as his focus point. They were able to demonstrate 12 microRNAs in urine that were significantly deregulated in patients compared to healthy controls. From these 12, they validated 2 and showed 1 that was expected to be a more general, non-tumor specific marker for cancer. Further on the endometrial cancer, in November 2020, a review was published regarding urinary biomarkers and their potential for the non-invasive detection of endometrial cancer. For endometrial cancer, early diagnosis has great potential to improve outcomes because of treatment can be curative, especially for early stage disease. However, current tests and procedures for diagnosis are limited by insufficient accuracy in some and unacceptable levels of invasiveness and discomfort in others. Again, urine is non-invasive, and urinary metabolites, proteins, and microRNAs have all been reported as possible endometrial cancer biomarker candidates. At this moment in time, there are just some limitations. There is insufficient evidence to support their use due to the lack and robust validation. And most of the studies exploring urine have been small pilot studies, and larger studies are needed. Several factors, including age, diet, level, physical activity, and medication use, should also be taken into consideration when people design studies for urinary endometrial cancer biomarker discovery. Next up, colorectal cancer, which already also has a lot of publications regarding biomarkers. One of them, Song et al., showed that total urinary DNA levels for KRAS mutations in colorectal cancer patients had a sensitivity of 19.7% and a specificity of 82%, and has shown to be useful for detection, treatment, monitoring, and prognostication. Furthermore, in another study, the same group showed that the hypermethylation of FIM was present in 85% of tissue samples and 75% of the paired urine samples, which represents an overall good correlation between tissue and urine samples. Hazou et al. Had a look at nucleosides, which are modified metabolic products of RNA and DNA degradation. And they were able to show that cytidine, 1-methyladenosine, and adenosine were significantly elevated in patients. With their study, they confirmed a study from 1977, which showed similar results. Urine is also seen as an attractive source for volatile organic compounds, this mainly because there is a biological pre-concentration, which occurs in the kidneys that reduces the signal-to-noise ratio. The overall performance of VOCs ranges from 63 till 93% sensitivity and 60 till 91% specificity. However, validation studies need to be performed to confirm these results. Next up, renal cancer. Several studies have been performed on single microRNAs and microRNA panels to investigate their use as a biomarker for renal cancer. Some showed a significant upregulation in urine samples of patients compared to healthy controls, and others were significantly downregulated in patients. But more interestingly is the fact that some of the significantly upregulated ones were downregulated in postoperative samples of the same patients. Promising for treatment response, you should think. But I would like to bring your attention to two specific publications. The first one from Pinheiro Pinto investigated microRNA 30A5-PME in different urinary fractions and was able to show a significantly upregulated in both urine sediment as supernatant of patients compared to healthy controls. And they were also able to demonstrate that this microRNA can discriminate patients with metastasis from patients without metastasis, with a high performance even. A second study from Nutsu et al., they were able to develop and validate a method for the detection of urinary cell-free DNA methylation in renal cancer patients. Campi et al., on the other hand, performed a systematic review of literature regarding renal cancer and showed 3 really promising articles for urine. Butts et al. Examined urinary cell-free and exosomal microRNA expression as a non-invasive marker. A microRNA combination was significantly differentially expressed between benign tumors and renal cancer. The microRNA ratio was also able to discriminate the tumors. A second study, Mitsik et al., evaluated urinary expression of microRNA 15A as a diagnostic biomarker. A statistically significant difference in median urinary levels of microRNA 15A was noted between patients with malignant and benign renal tumors, while on the other hand, there was no significant difference between the renal cancer subtypes. And third but not least, Liu et al. Assessed the urinary metabolome in patients with renal masses, applying a logistic regression algorithm. The authors used a metabolite panel consisting of L3-hydroquinurine, 1.7-dimethylylhumanazine, and tetrahydroaldosterone 3-glucuronate to establish a predictive model with an area under curve of 0.86, the sensitivity of 75%, and a specificity of even 80%. Let's now switch to the less obvious cancers for urine. Yamato et al. Performed a review of literature in October 2020 about a non-invasive detection of gastric cancer, and they also pointed out some studies around urinary biomarkers, among which was a study regarding microRNA 376C that showed elevated levels in the urine of patients with this cancer. Urinary microRNA 376C predicted gastric cancer with an area under curve of 0.70, a sensitivity of 60, and a specificity of 64, which are really promising results. On the other hand, Wang et al. Demonstrated the potential of machine learning and novel statistical approaches in aggregating multiple liver cancer biomarkers. This for the detection of a highly sensitive liver cancer screening test. Their prediction model combines serum alpha-fetoprotein and 3 urinary markers. These urinary markers were chosen for the development of this screening test because they were detectable in urine, which is a preferred sample type. More importantly, these urinary biomarkers are derived from different cancer pathways, thus providing the potential to overcome the heterogeneity of liver cancer. Next up, lung cancer. Several studies have been performed investigating the use of urine as a liquid biopsy for the detection of lung cancer. I first would like to highlight two results of some recent papers. The first one from Renal showed that the urinary cell-free DNA concentration of line 1 fragments of 97 base pair and 266 base pair were significantly regulated in patients compared to healthy controls, which consists of the findings of a previous study done in blood. Furthermore, they showed that the sensitivity and specificity of this biomarker can be increased by the combination of measurement of both fragments. On the other hand, Louis et al. Demonstrated that promoted DNA methylation in urine samples from COD1, TAC1, HOX-A9, and SOX17 could potentially act as a biomarker for lung cancer. They showed that significantly more patients with cancer has this promoter methylations compared to healthy controls. Furthermore, they also showed that the combination of the detection of these promoter methylations in both urine and blood could increase the sensitivity and specificity even more. A review from November 2020 by Urine et al. Showed promising results for non-small cell lung cancer detection in urine. Among others, they mention a study in 63 patients with advanced EGFR mutant non-small lung cancer. They detected sensitivities of 73% for tissue, 75% for urine, and 82% for blood. Excuse me. In the same study, a significant decrease in T790M MAF was found in urine from 9 patients who received treatment, highlighting a potential for follow-up of treatment response. These findings were confirmed in another study investigating ctDNA kinetics in 8 treated patients, from whom the early kinetics of ctDNA shed into the urine correlated with tumor response. Another prospective study in advanced non-small lung cancer demonstrated a good correlation and complementarity between the genomic profiles of cell-free DNA extracted from plasma, sputum, and urine compared to tissue, which is also a really potential promising results for screening purposes. A number of studies have suggested that urine could contain potential biomarkers for breast cancer screening, ranging from metabolome profiling and proteomic profiling to exosomic analysis. One study has demonstrated that 59 urinary proteins are differentially detected in breast cancer patients compared with healthy control individuals, and of them, 36 urinary proteins are stage-specific. A panel of 13 upregulated proteins could be used for breast cancer detection. Another study identified a specific panel of 4 urinary microRNA types that could discriminate breast cancer patients from healthy controls with a sensitivity of 98.6% and a specificity of 100%. Next to that, even urinary exosomes were investigated for breast cancer, and one study showed that the combined detection of microRNA 21 and metalloproteinase 1 had a really good overall performance for the diagnostic detection of breast cancer. There were even studies performed investigating the metabolome of breast cancer patients. One study showed the overall good performance of the combination of the detection of succinic acid and dimethylheptanoyl carnitine. So also really promising results for breast cancer. Next up, thyroid cancer. Hang et al. Showed that even thyroid cancer can be detected using urine as a sample type. Moreover, they used urinary exosomes as starting point for the detection of thyroglobulin. They were able to show that this urinary exosomal thyroglobulin can be used as a non-invasive, reproducible, convenient, serial, and accurate follow-up marker for patients with thyroid cancer. During their study, they also compared urinary exosomal thyroglobulin with serum thyroglobulin and showed that it can be an important pro-inflammatory predictor and biomarker of thyroid cancer recurrence for certain patients. The same group also has a clinical study ongoing according to clinicaltrial.gov. regarding the prognostic use of urinary exosomal biomarkers for thyroid cancer. Last but not least, essential methods such as calcium, magnesium, copper, and zinc are essential for life and play a fundamental role in a number of vital physiological functions. Due to their important roles, these essential metals are tightly regulated in the human body. But their dysregulation has been found in several cancers, among which pancreatic cancer. A study from Schilling et al. Showed for the first time that metabolomics is a promising approach for discovery of biomarkers for detection of pancreatic cancer, completely, non-invasively, using urine samples. They showed that significantly lower urinary calcium and magnesium concentrations are found in patients compared to healthy control, which seems to reflect a disruption of cell proliferation and protein synthesis affecting the calcium efflux and intake in the cell. Significantly lower levels of both elements have previously been reported in plasma, which is now confirmed in urine. Furthermore, they showed that patients exhibit a significantly higher level of copper and zinc than observed in urine of healthy controls. Increased copper levels have been previously described in both tissue and serum in patients, the former being sad but true, negatively associated with patient survival. The results of this proof of concept study suggest that metabolomics studies have the potential to be a source of new biomarkers for the detection and potentially monitoring of pancreatic cancer, completely non-invasively using urine specimens. However, larger studies are now warranted to determine if such analysis can help unravel early changes in PDAC development and ultimately improve the currently poor survival of PDAC patients. It takes time to develop a clinical biomarker. The process of development of a biomarker for early cancer detection is normally divided into 5 phases. Although many potential non-invasive biomarkers have been reported, most of them remain in phase 1 or 2. A few in phase 3 and 4, and sad but true, none in phase 5. Analytical validity of any biomarker has to be evaluated for accuracy, reproducibility, and reliability before they become a clinical utility. For the clinical application of biomarkers, the standardization of specimen collection, handling, storage, and analysis is key for quality assurance, reliability, and reproducibility. With the significant advances in analytical techniques and tumor biology, there is a great possibility of a more accurate, sensitive, and cost-effective non-invasive test for the early detection of cancer being developed in the near future. As you can see, there are promising first results on urinary biomarkers published so far, and some even turned into a test which received FDA approval. However, there are still some issues that need to be solved for urine as a liquid biopsy for cancer detection. Same issues like the field is solving for blood. There is a lack of uniformity and standardization. Optimized preservation and storage conditions are not known yet. Optimized isolation and detection techniques need to be determined. And of course, validation studies in larger cohorts need to happen. One of the main issues to be solved is the preservation of urinary analytes for cancer detection. So let's have a look at why preservation is so important. When urine is held at room temperature and left unpreserved, on the one hand, this can lead to microbial growth and lysis, and on the other hand, this can cause physical-chemical changes like pH, salt concentration or precipitation, protein concentration or precipitation, etc. These two major events have several consequences, such as the contamination of the extracellular space by unwanted microbial nucleic acids, which can have an effect on downstream analysis, such as NGS, the release of nucleases, cofactors, and pH changes that affect the chemical stability can cause degradation of cell-free nucleic acids. Furthermore, human cellular instability can occur, which in turn can also lead to the contamination of the extracellular space with unwanted cellular DNA and RNA molecules, again with a possible effect on the downstream analysis. And last but not the least, it can lead to the increased release of EVs and decrease of intact EVs. So overall, urine requires proper stabilization to lower the effect of collection and processing variability. Some of these issues can be solved by using a standardized and volumetric collection method, which is exactly what the Colibri platform offers. Next to that, our platform also provides the possibility to choose a preservation chemistry based on your purpose. Please, if interested, check out our booth at the EACR website or on our own website for more information, or contact me or one of my colleagues with your further questions. Then I would like to end by thanking all people involved from Novosanis, DNA Genotech, and of course my promoters and mentors at the University of Antwerp. And I would like to thank you for watching my presentation.